basically, I'm a trained veterinarian, but has worked as a microbiologist all my life. So there will be something with the practicals maybe I will not be able to answer questions on. So to begin with, Denmark, just to be sure you all know, is a small country in the north of Europe. We're only 5.5 million people. 1.2 of those people live in the capital of Copenhagen. The main livestock produced in terms of number is poultry, not shown here. Then at any time point, there'll be around 12, 13 million pigs alive in Denmark, while there's only 1.5 million cattle. And roughly 500,000 of those are dairy cows, standing in 3,000 dairy farms. So around 200 cows in average per dairy farm. So this presentation follows neatly onto the previous one. So it deals with Salmonella Dublin. So Salmonella Dublin is specific among the Salmonella types because it is adapted to cattle. So it's a host-adapted serotype, like typhi is adapted to humans. Though it can infect other species, and notably humans can get infected. So when you first see Salmonella Dublin in a herd, there will be clinical salmonellosis. It could be diarrhea in young stock. You can see depression, fever. You can see pneumonia as a sign, and you can see abortion in the cows. But as time progresses, the standard is that herds will be infected, but we'll really not see any clinical signs. Because it's a host-adapted, the pathogenesis is actually different from a standard Salmonella typhimurium infection of cow. It will go systemic, and it will locate at systemic size, and animals that have been infected can become carrier animals. For a long time, we thought that was very important for the epidemiology, but actually simulation studies in Denmark have shown that it does not drive the epidemic. It confers production losses in the herds, so that's also different from most other Salmonella serotypes. The farmer can actually feel that there is something wrong here in the first years after infection. It's not unusual that the margin drops considerably per cow stall. So if you're in the business of trying to understand what's going on with Salmonella Dublin, we've had the problem that it is a very narrow clone. So all the beautiful typing methods, molecular typing methods that are used to follow bacteria from one herd to another, they do not work very well with Dublin. They look too much like each other. They have some selective pressure to be like Dublin should be. So that's sort of where we start this out. You can detect Salmonella Dublin infection on herd level by an LPS ELISA. So using bulk milk samples or blood samples from animals. And of course, you can do traditional culture and you can do PCR. As I said, Salmonella Dublin infects humans. This is a list of the top 10 Salmonella serotypes in humans in Denmark in year 2014 and 2015. I have marked up Dublin, and you can see it's not a lot. It's approximately 2% of human cases in Denmark every year, but it's feared by doctors. So it has a very high case mortality rate, similar to listeriosis. So 30–40% of humans that get a Dublin infection will succumb to the infection. Mainly for that reason, we initiated a control program in Denmark. It started out as a control program, and then it grew into an eradication program where the initial goal was to eliminate Salmonella Dublin from cattle in Denmark, first by 2013, which did not happen, then by 2016, which did also not happen. So then we started to worry and thought, what is really going on here? So in this system, herds are allocated into level 1 to 3 based on antibody titers in milk and blood samples taken according to a very detailed and very complicated scheme, and you can ask me outside. It's not important for the overall conclusions. In this, level 1 are all the herds that are considered free from Salmonella Dublin. Level 2, they are the herds that have high antibody titers. Level 3 are the herds that, it started out as those are the herds with clinical salmonellosis, but now it is all herds where we have a positive culture, so a bacteria in the hand. They will be considered a herd where there's active spread of Salmonella Dublin. Right now, where it's an eradication program, what happens is if you're level 2, and you, as I said, you become level 2 if you have very high antibody titers, or if you trade with herds that are in level 2 and 3. Then you will be asked to make a mandatory planning of a reduction strategy together with your vet, and there will be intensified surveillance, and that will include culture. And if you are positive on the culture, then you move to level 3. If you're negative on culture, still positive on serology, you stay in level 2. Level 3 is basically under state control. So from there on, everything you do in that farm, you have to ask permission, more or less. But it will cost the farmer money in the sense that there will be hygienic slaughter, and the meat has to be heat treated. It cannot go to farmers or to consumers without heat treatment. There will normally be no selling of live animals, at least you'll have to ask permission. There'll be restriction on slurry application. And as for level 2, there will be your mandatory plan for reduction strategies. You can then test yourself out of being level 3 and move up to level 2, and then test yourself out of level 2 and move into level 1. And now, here's a very complicated slide, so let's take it easy. But it shows the percent of dairy herds, so milk-producing herds, found to be Salmonella Dublin positive from 2003 to 2018. And all the colors are different regions of Denmark. And if I went into detail, you would see that many regions are free from Salmonella Dublin. But this is the average of the country, so you can see started out around between 20 and 30% of dairy herds positive, milking herds positive, and a quite steep reduction. And then we ran into what you normally run into in such programs. There's a tail. And if we looked at that tail, it's a bit like the presentation on TB in Devon. They appeared to be herds that had been positive for a long time in serology. Now, as I told you, typing methods were not very good. If we sort of claimed that these farms were positive for a long time because they were doing something wrong internally, there was no way you could prove that they were not reinfected constantly from outside. Along came whole genome sequencing. So it's become feasible to sequence bacterial isolates during a routine typing investigation. All Salmonella isolates from humans in Denmark are actually subjected to whole genome sequencing now. So we took this method, and then we tried to answer two questions. One, we knew these herds with long-term C-reaction seemed to be the problem. So we wanted to answer the question, are they persistently infected? In which case, the reduction strategy should really focus on internal biosecurity. Or were they constantly reinfected from outside, in which case a program should focus on in external biosecurity. And then we wanted to see whether human cases of S. Dublin were domestically acquired or travel associated. Very short about materials and methods. We did this as a retrospective study. We collected Dublin isolates from the National Surveillance Program, from different research programs, and from human cases. We're talking about 197 isolates of Salmonella Dublin, and they're not a random sample. They were selected so that they represented persistently infected herds, and in this study that means a herd that was culture positive and remained seropositive for more than 3 years after the first culture. And we also included for control some non-persistently infected herds. Those herds has been positive once, then quickly serology went down, and they were negative for the next 3 years. They covered the year span from 1996 to 2016. And then on top of that, we included 46 human isolates from Denmark. They were only from these 3 years. And then we went on the net and collected sequences that were just published there from other countries. Then we did a full genome sequence, and then I said here, you can ask me about the methods outside. It's not important for the understanding, but the way we analyzed it is we did two things that maybe you should understand that. We did what is called a core genome SNP analysis, a phylogeny analysis, and a core genome MLST. And SNP is single nucleotide polymorphism. So in that, you defined yourself, this is my reference, and then you take all your isolates and see where do they differ. And based on this difference, you can say how much are they related to each other. The core genome, that's what they all have in common. So when you do this analysis, you ignore some information. MLST, multilocal sequence typing, is looking, instead of looking at single nucleotides, you look at genes. So alleles, if they have the same gene type on gene 1, then I give that a score 1, both of them. And if they differ in number 2, then they will differ on that. Okay, then. On to results. So the first result. So this is a SNP analysis. So this is phylogeny of Salmonella Dublin from cattle in Denmark. It's first thing you should notice is there's only a bit more than 1,000 SNPs in this, even though it spans from 1996 to 2016. To give you an idea of how unique that is, if we've taken typhimurium from humans, all typhimuriums from humans in Denmark over just 7 months, and they have 10 times more genetic variation. So this is very little. Color-coded according to, it's the western part of the country that is still positive, color-coded to geography. What you immediately can see is that we have a cluster here, we have a cluster here, and we have a huge cluster here. So what we think was a Danish epidemic spanning over many years appears to be 3 sub-epidemics. That's conclusion number 1. Then if you look at these, there's a lot of details hidden here that you cannot see. So now I will enlarge something that comes from up here somewhere. So this is just a small subset of what you see over here. So this is one farm. The farm is called VV1 in the, so VV is it called, and this is isolate number 1. It dates from 2001. This is isolate number 5. It dates from 2014. And what you see is there's no other herds mingled in here. These isolates from the same herd over 14 years span, they are the closest relatives of each other. So the obvious conclusion from that is they've had the same infection for 14 years. See, this, to be very sure that this was really true, we did an MLST core genome MLST, and here I have color-coded farms according to farm. And again, you can see farmed clusters. And actually this analysis, we dare not use it this way still, but it also tells us that this farm most probably is mostly related to that isolate. So you can start thinking also about transmission routes. We also asked the question, these persistently infected herds, is that because they have a particular nasty kind of doubling that has an ability to form biofilm or whatever in the herd? So this is the same as this, And here I've just color-coded according to whether it's a persistent herd or not a persistent herd. And I think I can convince you they mix in. So we're very sure that there's nothing particular about persistent Salmonella Dublin infection in terms of the bacteria. Maybe something very particular about the herd. The human thing, a little bit more complicated, because here we've taken in a lot of gene sequences from other places in the world, but this is the small Danish cattle cluster. This is the second one, and the big one is over here. And this, green is human isolates. The reddish one is cattle isolates. These are Danish human isolates. And if I enlarge that so you can see something, then what we see is that the majority of Danish human isolates, they cluster in with Danish cattle isolates. But we haven't found somebody that are exactly identical, but I don't know if we should expect that. So conclusions from this is the Danish epidemic with S. Dublin contains at least 3 sub-epidemics. The majority of long-term infections in Danish cattle herds is caused by persistent infection with the same strain. Improvements in the eradication program should thus focus on internal biosecurity. The majority of Danish human cases originate from Danish cattle, and it's most likely beef because we pasteurize all the milk. And whole genome sequencing significantly improved power of molecular typing of Dublin. And here I must add, I'm actually convinced that whole genome sequencing is one of the best things that happened to us, because I work a lot in developing countries as well, and this is a technique they can do just as well as we can, so they can catch up very easy instead of very expensive serology-based methods. And then just acknowledging my co-workers, Ekle, Lisa, Leonardo, and Shahana from my own university, Erik Rattenborg from the Danish Cattle Association, and Mia Torvedal and Gitte Sørensen from the Danish CDC, Statens Serum Institute. Thank you for your attention.